Alright everybody, welcome to the next video lecture. This is on nucleic acids. So this is wrapping up this week's material for the next exam. So you've got carbohydrates, you've got lipids, membranes, nucleic acids. I lied, this is not the last lecture. We've got one more after this. We've got replication. But DNA, this is the start of the DNA topic, which will wrap up the day and wrap up the week. Okay, so let's see. We'll begin with DNA of the nucleic acids, excuse me, as opposed to RNA. So DNA is the hereditary molecule. It's got the hereditary information in it, and it gets copied and passed on from generation to generation. Let's see. We don't need to really go over when it was discovered. I think you guys know. The structure of the double helix was most famously elucidated by Watson and Crick. I think they used X-ray diffraction combined with some other observations that were made by those two. And so we've got, this is the pretty familiar DNA structure here. Let's see, probably one of the most significant finds of the 20th century, the discovery of DNA. Anyways, the double helix, it's made up of a pair of DNA strands. Has at its core bases, which are joined together, and you can see those 4 different bases here. We'll talk about them in more detail. Adenine always pairs with thymine. Guanine invariably pairs with cytosine. The 2 hydrogen bonds are formed between adenine and thymine. You can see those here. And then 3 hydrogen bonds forming between guanine and cytosine. So that holds our double helix together. The complementary structure immediately suggested to the scientists that discovered this how DNA might be replicated. And it further explains how information in DNA is transmitted to RNA for the synthesis of proteins. In addition to the hydrogen bonds between the bases of each strand, the double helix is actually held together by hydrophobic interactions of the stacked nonpolar bases. And I don't know if I included an image of the base stacking here, but your book has a really nice image of the base stacking. So these nonpolar bases will stack. Crucially, the sequence of the bases in DNA will carry the information for making proteins. And we read these in groups of 3. We call these codons. Each 3 nucleotides will then make a codon on our mRNA strand as when we get into that translation and transcription next week. You guys will see more about that. But ultimately, those codons encode for proteins in that they code for amino acids. So each codon is an amino acid. An entire coding region or a gene codes for a protein's primary structure, the sequence. Okay, so let's talk about, try to give you guys keywords here. Let's talk about structure. So DNA strands are polymers of nucleoside monophosphates that are held together by phosphodiester bonds. 2 such paired strands make up the DNA molecule, which is then twisted into a helix. The most common form is the beta form, and I'm going to show you a few different images of that coming up. Most common form is the beta form, and this has the DNA helix repeating 1 full turn every 10.5 base pairs, with the sugar and the phosphate part of the molecule forming the covalent phosphodiester backbone. I don't know if this has a better picture of the backbone. It does not. But this would be backbone, and this would be base pairs, or nitrogenous bases that are going to do the pairing. Okay, so the adenine, the guanine, the cytosine, the thymine, these bases are oriented in the middle, where they will form their familiar base pairs and look like kind of like the rungs of a ladder as you're kind of climbing a winding staircase. Now, these make up the building blocks. These are the building blocks of both DNA and RNA. No, carrier settings. are you telling me that? Sorry. So, building blocks. So these are the building blocks of both RNA and DNA. So before we can talk about those different groups, we need to define some terms. So let's define some terms. Nucleotides contain three primary structural components. You can think of the word tide there, tide, as being the beginning of three parts. Nucleotides. So nucleotides have three important parts. They have a nitrogenous base, a pentose sugar, and at least one phosphate. Now, molecules that contain only a sugar and a nitrogenous base, or no phosphate, are called nucleosides. So, the base and a sugar. The sugar can be of two different types. The bases, you can see here, can be of 5 different types. Now, the nitrogenous bases that are found in nucleic acids include adenine and guanine. These are called the purines. Cytosine, uracil, and thymine, or called the pyrimidines. There are 2 sugars found in the nucleotides, deoxyribose and ribose. Deoxyribose is missing an oxygen, where ribose has an oxygen. So on the deoxyribose, there's just an H there on the number 2 carbon. By convention, the carbons on the sugars are labeled from 1 to 5 going in this direction around here. And so that's where we're going to get the number 5 that we hear all the time and the number 3 that we're going to hear all the time when talking about DNA. Deoxyribose differs at the number 2 position. Nucleotides containing deoxyribose are called deoxyribonucleides. Deoxyribonucleotides. And they are found in DNA. Nucleotides containing ribose are called ribonucleotides, and they're found in RNA. Both DNA and RNA contain nucleotides with adenine, guanine, and cytosine. So, adenine, guanine, and cytosine are found in DNA and in RNA. RNA contains uracil nucleotides, so RNA only. Whereas DNA contains thymine nucleotides only. That doesn't mean that the others don't exist. They do exist. They are just very uncommon. So when a base is attached to a sugar, the product is a nucleoside and it gains a new name. So uracil-containing nucleosides are called uridine if they're attached to ribose, or deoxyuridine if they're attached to deoxyribose. So uracil becomes uridine. Don't worry about the names too much. Thymine becomes ribothymidine or thymine. Thymine becomes ribothymidine or thymidine, depending on if it's attached to RNA and DNA respectively. Cytosine becomes cytidine or deoxycytidine. Guanine becomes guanosine or deoxyguanosine. And adenine becomes adenosine or deoxyadenosine. Now of these, the deoxyuridine and the ribothymidine are the least common. So that's the DNA version of uridine, or of uracil. The deoxyribose containing uracil is called deoxyuridine. It's one of the least common nucleotides, as well as ribothymidine. So the addition of one or more phosphates then to a nucleoside rounds this out and makes it a nucleotide. So, these are all nucleosides with phosphates on them. These would all be nucleotides then, nucleosides containing either monophosphate, diphosphate, or triphosphate. The number of phosphates in the nucleotide is identified or indicated by appropriate prefixes like mono, di, tri. Nucleotides are often referred to as nucleoside phosphates. Sometimes you hear that. That's because they are nucleosides with phosphate groups on them. Okay. Now, cytidine, for example, refers to, when you say cytidine, it refers to the nucleoside, no phosphate. But cytidine monophosphate refers to, I think this is cytidine right here. So this is cytidine. Cytidine is the name of the nucleoside, like what's pictured here. So just a sugar, just a base. You should be able to look at a sugar like this and know if it's DNA or RNA by looking at that position right here. This has a oxygen. This is a ribose, not a deoxyribose. So this would be an RNA. This nucleoside would be found in RNA. Okay. Anyways, and then when we add a phosphate group, it would be called cytidine monophosphate. Or if we were to add 3 phosphate groups, it would be called cytidine triphosphate, or CTP. Very similar to energy or the energy carrying molecule ATP, if you remember that. So let's talk a little bit about energy sources here. I can't spell. Okay, so ATP is the most common and best known energy source. Each of the 4 nucleotides, though, that we've talked about plays an important role in providing energy. So in addition to ATP, which we already know about, GTP, for example, is the energy source for protein synthesis during protein translation, or translation as we call it. This is taking the mRNA and making a protein strand from it. GTP is the energy source, as well as for a handful of metabolic reactions. A bond between UDP and glucose makes UDP glucose. This is a building block for making glycogen. Glycogen being, if you remember, the energy storage molecule in our bodies. CDP, so this would be cytidine diphosphate, is similarly linked to several different molecular building blocks important for glycerophospholipid synthesis. So some of the lipids that we talked about in the last chapter. The bulk of the ATP, as you know, or maybe you don't know yet, is not made directly from, the bulk of the ATP is not directly, what am I trying to say? The bulk of the ATP made in the cells is not directly coupled to metabolism, but rather by combined processes of electron transport, oxidative phosphorylation that occurs in mitochondria, and or photophosphorylation if we're talking about plants. Triphosphate energy, in terms of ATP, is transferred to other nucleosides or nucleotides by actions of enzymes called kinases. So what I'm saying is that the most of the ATP in our bodies is not just for metabolism, not just for breaking down molecules or building up molecules. A lot of it is there to provide phosphate groups. So not exactly there for energy or to drive, energy non-spontaneous reactions, but to be a source of these phosphates for us. We see that phosphates are required for creating some of these different nucleotides. Okay, now specifically, let's switch gears and talk about DNA. So, deoxyribonucleic acid. So, individual deoxyribonucleotides are derived from corresponding ribonucleoside diphosphates, meaning we make DNA from RNA molecules, or from RNA sugars. So we have to start with a ribo sugar to make a deoxy sugar, or a deoxyribo. The deoxyribonucleoside diphosphates are then converted to the corresponding triphosphate by the addition of a phosphate group. So we start with a ribonucleoside diphosphate. So that could be something like, let's just use uridine. So it would be like a UDP. Well, maybe that was a bad example, because that one, we don't make DNA out of that one. So we would start with an ADP, so an adenosine diphosphate, or how did we call it when it was a ribo? This would have been an adenosine diphosphate. It's called deoxyadenosine when we're going into the next. So this is adenosine diphosphate, and then it would get turned into deoxyadenosine diphosphate. So this is RNA, and then DNA, and then it would get turned into adenosine triphosphate. So RNA to DNA, and then to the nucleotide triphosphates. The only one that doesn't go through this pathway is thymidine, because as I mentioned earlier, RNA versions of this guy, thymidine diphosphate, are very rare. Ribothymidine. So we're not going to go over that, the how it is made. If you're interested, you can Google that. Sorry if I teased you. If I thought I was gonna give you the answer here. Just noting that most of our DNA molecules are made from RNA molecules, but not thymine. All right, building DNA strands. So let's put together. Let's polymerize this guy. So building strands. All right, each DNA strand is built from the DNTPs, so the deoxynucleotide triphosphates. It's just a general term for generic, ATPs and GTPs and whatnot. So each strand is built from these by the formation of a phosphodiester bond that's catalyzed by DNA polymerase. Now the phosphodiester bond is carbon number 5 forming a phospho. Well, no, that's the bad start to the. We're going to look at this phosphate group linking two nucleotides. So we've got a 3' phosphate on this nucleotide that's attached to the 5' carbon of the next. I guess this is still one nucleotide, two nucleotides, but they're linked together by this phosphodiester bond. So one of the ester bonds is here, one of the ester bonds is here. It's a phosphoester bond because that's not a carbon, and so this is where the name comes from. So this is a phosphodiester bond. This is catalyzed by DNA polymerase that we'll take a look at later on. And again, between the 3' OH of one nucleotide and the 5' phosphate on the next nucleotide. So as you can see, on the 3' end of this growing strand, we've got a thymine, and this thymine here is attached to a deoxyribose sugar that has a free hydroxyl group on its 3'. So the next nucleotide, which you can kind of jump up here and kind of see what that might look like, would be a nucleotide that comes in with a phosphate group attached to its number 5, but nothing else attached to the phosphate group. And so that phosphate group is going to form another ester bond here at this hydroxyl, linking the two nucleotides, and then that nucleotide will have a free hydroxyl at the bottom. We often designate, so because of this, the way that they attach, we say the directional growth of the strand is from the 5' phosphate side to the free 3' hydroxyl side. So DNA grows from 5' to 3'. This is how DNA gets polymerized. And so when we talk about DNA polymerase, we're often going to say that it has 5' to 3' activity, meaning that that's the direction that it is catalyzing these new bonds and making a polymer. Okay, so you can see two strands here. We've got one strand with the 5' side up at the top, one side with the 3' side up at the top. These are complementary to each other. So you can see the other thing I want to point out here is that they're anti-parallel to each other. So they're parallel in that they go the same direction, anti in that they run in opposite directions. So we've got a 5' side that as we go this way, we would read it as T, C, G, A. And we can see then if we wanted to read the complementary side, we'd have to start at the 5' and read this way, and we would read T, C, G, A. It's the opposite. It's the opposite. Because of base pairing, a T becomes an A, a C becomes a G. So we're going to get a complementary strand here that is also anti-parallel. All right, they're held together by hydrogen bonds. So hydrogen bonds between the base pairs hold the nucleic acid duplex together. So 2 hydrogen bonds per adenine and thymine, 3 hydrogen bonds per guanosine and cytosine. The B form of DNA, which we haven't looked at yet, has a prominent major groove. Here we go. So I've got two versions of this. I've got a ball and stick sort of space filling model, and I've got a more of a schematic here, so you can see large groove, small groove, and then here again major groove, minor groove. So these two should correspond to each other. Now the prominent major groove and minor groove sort of trace the path of the helix. You can kind of see that. Proteins such as transcription factors might bind to these grooves and access the hydrogen bonds of the base pairs to read, quote unquote, the sequence. Now there are other forms of DNA that are known, and I've got those shown here. So there's the alpha form, there's the beta form, and there's also, alpha, beta, there's the B DNA, there's A DNA, and then there's Z DNA. So this here is A, this is B, this is Z. Now, a little bit about these. So bDNA is considered to be the normal physiological DNA. Right-handed helix, this one has the 10.5 base pairs per turn, kind of a normal DNA. We have aDNA. This is also right-handed, but it's a little thicker in terms of the distance in between. There's also 11 base pairs per turn on this one. And then we've got the zDNA. This one is different. This one's left-handed, so it turns the other way. It may play a role in the regulation of gene expression. That's one thing people think. It occurs in nature usually when we have alternating purine and pyrimidine bases. I don't know if I included it. I did. So alternating pyrimidine and purine bases. So like GC, 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 having that kind of sequence has a propensity for making these structures happen. They are produced by flipping one side of the backbone 180 degrees without disturbing the backbone bonds or the hydrogen bonding. So I don't know that this is exactly very easy to picture. I'm sure there's a video on YouTube that I probably could have dug around for that may have shown that a little better. I apologize for not having that video for you. All right. Anything else about the Z? Oh, the Z form sort of has a little bit of a, if you can kind of picture it looking back at the Z one, a little bit of a zigzag shape. This is why it's got a zDNA name. It's a little more stretched out as well compared to the A and the B forms. Okay, so let's talk a little bit about how why this zDNA form actually forms by long stretches of G's and C's, and there's a little bit of a connection to tension and superhelical tension here that we want to get into. So let me get my slide. Okay, so short stretches of the linear DNA duplexes actually exist in the beta form. Double helices of DNA in the cell vary in the number of double helices in the DNA in the cell can vary in the number of base pairs per turn that they have. So we talked about beta form being normal, talked about beta form having, actually I was going to put we talked about beta having 10.5 base pairs per turn. Okay, so normally during regular processes in the cell, double helices can actually have more base pairs per turn than 10. Several reasons for this. So, for example, during DNA replication, strands of DNA at the site of replication get unwound at the rate of about 6,000 rotations per minute by an enzyme called helicase. And we'll look at helicase a little more when we get to replication. Now, the effect of such a local unwinding in one place in the DNA has the effect of increasing the winding ahead of it. So, unrelieved, that tension in the DNA duplex can actually result in structural obstacles to the machinery that does the replication. So, there are adjustments that can occur in a couple different ways. So, first, the tension that's being produced here can actually provide energy for flipping DNA structures. So zDNA can actually arise as a means of just relieving the tension. So that might be why there are stretches of those GC, so that during replication, when, and we'll discuss this image in a second, when the DNA is getting kind of super wound up, these zDNA stretches can form where this backwards helix forms to relieve the tension. Second, DNA can actually supercoil to relieve some tension, and I've got some images that I'm going to show you regarding that. One of them is actually, they're both here, so you can see in this is a prokaryotic genome, so it's circular. Depending on which way you, either if you negatively supercoil or positively supercoil, that just means twisting it, right, without cutting or untying anything, just twisting. If you've ever done this, played with a rubber band, you know this happens. We can get positive and negative supercoils, and we actually have molecules in our bodies called topoisomerases that do these jobs. We'll talk about topoisomerases here in a second. Gyrases, reverse gyrases, also proteins that will twist or turn DNA. Here you can see electron micrograph images of some DNA, probably from a cell. I don't know if these are all within the same cell or different times of that DNA, but you can see the relaxed circle, you can see a linear portion, you can see some very supercoiled portions. Okay, so supercoiling can also relieve tension during the spreading apart of the unwinding of this DNA helix during replication. We'll talk a little bit about how that happens in a second, and then again, enzymes called topoisomerases can do the job. So I want to talk a little bit about the topoisomerases. So circular DNA actually has this happen more often. Double-stranded DNA where the 5' and 3' ends are just joined by a phosphodiester bond. Bacteria, prokaryotes tend to have circular DNA. And then it can have negative or positive supercoils. What I'm showing you here is actually something called topological isomers, and there are different parameters. We're not going to go into that. I just wanted to show you some of the twists and writhes. So twists and writhes that are possible with DNA duplex coiling. Okay, so lots of different little structures can happen, but let's focus on this side for a minute. So as far as this goes, we have, well, in bacteria, there are enzymes called gyrases. I don't know if we exactly have gyrases or if we have helicases, but DNA gyrase, for example, is a bacterial topoisomerase. Now, a topoisomerase is an enzyme that can relax supercoiling in circular DNA. There are two classes. There's class 1. Class 1 topoisomerases will cut the phosphodiester backbone of one strand and then pass the other strand through and then reseal it. Class 2 cuts both strands, kind of loops the DNA over itself and then heals it or reseals it. And so you can see here in this circular loop how this DNA gyrase might kind of come in here, cut a strand, loop it over, reseal it behind. So here it looks like it's in front of the DNA, it gets cut, and now it's behind the DNA. So we've got this sort of, and it can do that over and over to introduce supercoiling. See, coming back here. And again, there are reasons why we want the DNA to be able to get compacted, right? Because there's a lot of it, and we need to fit it all in our cells. Let me see where I am here. Okay, so we're going to talk about packaging now. I think there was one other. Obviously, I want to talk here about some of the unwinding. So let's talk about denaturation. So just like proteins can denature, DNA can denature also. And so energy actually has to be added to a sample of DNA to break the hydrogen bonds and to disrupt the stacking, the base pair stacking. This is carried out by heat. You can heat a DNA solution. Heat denaturation of DNA is called melting. And so you can see here, well, you can't really see it here, but we call it melting. And we often talk about the melt temp, or the TM, which is the midpoint of a melting curve. I think I might have a picture. I don't have a picture of a melting curve. I think we talked about a melting curve on something before. Maybe I'm thinking of another class. Okay. As the DNA gets heated, the strands will separate. We can measure this in a lab and actually calculate that melting point by using light absorption. So DNA bases absorb light in the 260 nanometer wavelength. As the DNA unwinds, the absorption doesn't change, but the amount of light absorbed changes. So twice as many DNA strands, right? Where we had one DNA strand or one double helix, it unwinds, and so we've got twice as many kind of molecules, so it absorbs more. And so they can actually look at that. They can plot the temperature versus absorbance, and they can find like a midpoint or a transition point. And anyways, DNA that has higher percentages of CG has higher melting points because they have more hydrogen bonds, 3 hydrogen bonds per GC compared to 2 hydrogen bonds per AT. So in the old days before sequencing, they used to just find the melting points of different DNAs and compare them that way. It gives you an idea of the GC content versus the AT content. Not informative. We have about the same GC content as E. coli, but clearly we are not anything like E. coli. So until sequencing came around, that was all you could really do, is kind of compare those things. Okay, and so these images just show as we apply heat. Now, what's neat is that DNA will actually renature if you let it. So if you remove the heat and you just let these strands kind of commingle, they will form up again. And this is the basis of the technique called PCR, polymerase chain reaction, which is how we amplify DNA. So you need to also have some primers that are specific. They're RNA primers that we'll learn what primers are important for when we do replication in the next lecture, but you need to have some primers in there for the DNA sequence you're interested in. You got to have nucleotides, deoxynucleotide triphosphates. You got to have a bunch of those, and then you just have to have a way of maintaining temperatures or changing the temperatures. And what you do is actually you need one other thing. You need something called Taq polymerase. Taq polymerase is a enzyme that, so just like our polymerases will make DNA polymers, Taq polymerase works at high temperature. We've taken this out of some kind of bacteria that lives in hydrothermal vents or something hot, a hot lake or something like that, hot springs. It doesn't denature. It's a protein that can stand up to high temp. So we take that. And then we subject DNA through a series of heating, cooling, heating, cooling. We heat it so it splits apart. All of these then go and make copy strands on each of the now separated strands. And so then, we have 2 sets of DNA. And then we heat it again, and those split, and then we double those, and then they condense again, and then, now we've got 8 strands of DNA, and then we heat them up, and they split again, and then we copy everything that's open. That's called polymerase chain reaction. Every cycle you do, you double the amount of DNA you have. So very, very quickly, you can take one strand of DNA and have over 1 million strands of DNA, or over 1 million strands of the gene that you're interested in. Okay, so denaturing and renaturing as part of what DNA can do. All right, let's talk about how DNA is packaged and contained in the cell. Packaging. All right, so while DNA is easily the largest macromolecule in the cell, single chromosome of a small bacterial cell, for example, can have a molecular weight of over 1 billion Daltons. I don't know if you guys are familiar with the unit of Daltons. Um, if one were to take all the DNA of a human chromosome from just, well, actually from all of your chromosomes from just a single cell and lay them end to end, it would be over 7 feet long. Like 7 feet long of a DNA, of all your DNA, from your 23 chromosomes all stretched end to end from one of your cells. That's a ton. So it's an enormous molecule. It demands careful packaging to fit within the confines of the nucleus, or inside of a tiny bacterial cell. So the chromatin system of eukaryotes is the best known, but bacteria have a system for compacting DNA too. Let's look at DNA in bacteria first. So bacteria, there's no nucleus. So instead, DNA is just contained in a structure called a nucleoid. It contains about 60% of DNA, with much of the remainder comprised of RNA and transcription factors. Bacteria don't have histone proteins that wrap around, that DNA wrap around like we do. But they do have proteins that help organize the DNA in the cell, and mostly by making sort of these looping structures. So this is the nucleoid. The proteins are called nucleoid associated proteins, and include ones named, actually I don't remember what they're called, HU and other weird things, CBPA. Anyways, of these, HU I remember because it's the most, it resembles the most like the eukaryotic histone H2B, and binds to DNA sort of non-specifically. It just, the charges on these proteins are often very positive. They contain lots of positive amino acids, so that DNA, which is very negatively charged because of all those phosphates, they just stick together. The proteins associate with the DNA. They also cluster. That might be the origin of the kind of loops that they see. And it's likely that these proteins play a role in helping to regulate transcription, which is the I don't know what word I'm trying to find. It's the taking the genes and making proteins from them. The expression. There we go. Transcription regulates the expression of the genes. Okay, so on that note, let's talk about eukaryotes. So we know a lot more about eukaryotes. So the method that eukaryotes use for compacting DNA is considerably different. So eukaryotic DNA is typically much larger. It fits into the nucleus that's not much bigger than the prokaryotic cell. So it's much bigger and it has to fit into the same space. Human DNA is about 1,000 times longer than prokaryotic DNA. So the strategy employed by the eukaryotic cells is that of spooling. So DNA is coiled around positively charged proteins called histones. And so you can see this is a few different histones here. There's a histone core that's made up of some octamers of 2 H2As, 2 H2Bs, 2 H3s, and 2 H4s. 1, 2, 3, 4, 5, 6, 7, 8. There's 8 in there. And then there's histone 1, which is not found kind of clustered around these guys. They're normally separate. So you've got all of these proteins, all these little positively charged proteins called histones. Here in this other picture, you can see the same thing. This is another histone. In gray, you can see the DNA wrapped around it. Just like you see this in red is the DNA wrapped around these histones. The different colors that you're seeing are the different subunits of this histones. So these proteins, whose sequence is very similar in cells as diverse as yeast and humans, so different types of eukaryotes, completely very similar histones. They come in 4 types. There is a 6th type that's referred to as H5. It's actually just an isoform of H1. It's rare, but it isn't an actual 6th type. It's just another variation of one of the H1 proteins. But anyways, there's an octamer found in sort of the core of these little histone complexes, and this is the fundamental unit of chromatin. We call it the nucleosome. So you can see some nucleosomes right here. A core of 8 histone proteins with DNA wrapped around it form a nucleosome. Now, the proteins themselves are an octamer, and I think the DNA that wraps around the octamer totals around 147 base pairs and makes 1 2/3 turn around it. So if you're into those specific details, its complex is referred to as a core particle. There are linker regions, and so you can see this sort of linker region here, about 50 to 80 base pairs that separate core particles. So nucleosome is a core particle plus a linker region. Histone H1 sits near the junction of the incoming DNA and the histone core. So that's what this green guy's doing here. It's often referred to as the linker histone. And in the absence of H1, non-condensed nucleosomes resemble beads on a string. So this is to kind of show you what the electron micrograph looks like of beads on a string when the H1 protein is missing. H1 protein would be kind of holding all of these guys together. And then here I've got some sequences for you. So this is the histone protein. They're all similar in structure, and they're all rich in basic amino acids. So you can see lysine and arginine shown in red. These are positive at physiological pH, so they form tight bonds with the negatively charged phosphate backbone. All right. Now, for DNA, compression comes at different levels. So the first level is at the nucleosomal level. Nucleosomes are stacked and coiled together into higher order structures. So these 10 nanometer fibers are the simplest higher order structure. Those are the beads on a string. They grow in complexity. 30 nanometer fibers consist of stacked nucleosomes packed together, and then higher level packing produces the metaphase chromosome that you find in meiosis and mitosis. And we'll talk about some of the cell cycle a little bit too in the next lecture. So this chromatin complex, it's kind of a logistical concern for the processes of DNA replication and particularly transcription or gene expression, where specific regions of DNA must be transcribed. Altering chromatin structure is therefore essential for transcriptional activation. It's important for being able to let the proteins that associate with DNA get to all the different parts that they want to get to, all the different genes that are wound up around histones that need to be transcribed, need to be made available. So one strategy involves adding acetyl groups to the positively charged lysine side chains of the amino acid histone amino acid to loosen their grip. So acetyl groups aren't going to have the same charge on them, so they'll lose their grip. And this allows proteins involved in activating transcription to gain access to the DNA. Let's talk about mutation. Let's see how many slides I have left here. Oh, we got to talk about RNA also. Okay, so mutation. So 2 topics. We've got the Ames test. Both of these are showing some mutations. So first I want to talk about mutations and mutagens and kind of how common they are. And then we'll look at what we see in terms of mutation rates along the histone kind of beads on a string, kind of make some comparisons. So the Ames test is a way to compare if a molecule causes mutations in DNA more than normally would be caused by just random nature. Basically to decide if a compound is a mutagen. And it's named for the guy who invented it or discovered it or who first created the test. It's an easy test. It starts by taking an organism and making a gene in that organism that's non-functional. So for example, a strain of Salmonella can be created that lacks the ability to grow in the absence of histidine. So it would normally be able to make histidine, okay? But you take the gene that allows it to make histidine and you break it. So now it doesn't work. So now, if you don't provide histidine, the Salmonella will die. So without histidine, it'll die. So then what you do is what you look for is if that histidine gene comes back. And so you can take Salmonella that can't grow histidine or can't grow in histidine and grow them with histidine. You put histidine in there so they grow. So they're all happy. You split them. You put half of them into a plate, an agar plate for incubation, with the compound that you want to test its mutagenic properties. You take the other half of those histidine requiring salmonellas and you just plate them on a plate. And both of these plates have no histidine on it, or very little histidine, so they shouldn't be able to grow. Now then you wait. Now in both cases, you're going to get some growth. You're going to get some strains, some cells in here that actually mutated back to working again. So you took the gene, you changed an amino acid so that it wouldn't work, and then we just see, what might change that amino acid back and make it work. Now, there's a whole lot of other mutations that could happen, but we're looking for one specifically, and we're using that to kind of gauge, because even you can even see here in the actual no mutagen containing sample, some of those salmonellas mutated into a way that allowed them to make histidine again, and they were able to make it without histidine in the media, and they were able to grow. That's how nature, finds a way, right? Mutations are normal, and mutations often bring back activity that might have been lost. So out of all of these salmonella that went into this plate, only 2 mutated, and those 2 eventually gave rise to little colonies. That's really cool. In the mutagen side, though, you see that there was a lot. So this indicates that this compound here actually is pretty bad. It's a mutagen. If we were to see around the same number of colonies in both plates, well, then that compound might not have been a mutagen, at least not one that's more dangerous than the normal mutation rate. Okay. Mutations do happen at low levels in cells, completely normal. Let's talk a little bit about what we see in terms of the position along that kind of beads on a string. So what you see here is that the spacing of the nucleosomes is somehow important to mutation. So here, the DNA that's wrapped around these histones has a very low frequency. Okay, actually, let's look at the 2 different types of mutations that are happening here. We have 1 base pair insertions or deletions, more than 1 base pair insertion or deletions, and substitutions. Now, of course, insertions or deletions are really bad because that changes the reading frame. Your 3-letter codes now all shift over by 1, and now not a single one of them is going to be correct anymore. Whereas a substitution messes up 1 set of 3-letter codes but leaves every other 3-letter code okay. So we see here that DNA that's wrapped around histones has a much higher frequency of a mutation by substitution than by a mutation by insertion or deletion. And that in the regions where DNA is not wrapped around histones, that there is a lower substitution frequency and a higher insertion or deletion frequency. So somehow this means that these linker regions or these nucleosomes themselves are have some way of changing the mutation rate or modulating the types of mutations that happen. So again, mutations are normal. They're also not constant around the nucleosomes, and I thought that was interesting. Okay, let's talk about RNA. So we have a few differences here with the RNA. RNA for one is single-stranded compared to the double-stranded, but we'll get there. So the structure of RNA very similar to that of DNA. Similar to that of a single strand of DNA, but the ribonucleotides obviously they're joined together by the same sort of phosphodiester bond. Uracil is going to take the place of thymine in RNA, so we're not going to see thymines. RNA is assembled by RNA polymerases, which copy DNA templates, much in the same way that DNA polymerases will make DNA strands copying DNA templates. During the synthesis of RNA, uracil is used across from adenine in the base pairing, different from in DNA where we see thymine pairing with adenine. The building of messenger RNAs, so mRNAs, by copying a DNA template is crucial in the step of transferring the information from DNA into a form that directly synthesizes or that directs the synthesis of a protein. Additionally, ribosomes and transfer RNAs, so rRNAs and tRNAs, serve an important role in reading the information in the mRNA and in polypeptide synthesis. So RNAs are also known to play important roles in the regulation of gene expression. Now, normally, or the central dogma of biology has been that DNA is the information carrying molecule, and that it goes to RNA for translation and making into proteins. What we've kind of seen in the last couple of decades is that that's not always true. There are retroviruses. These are viruses that don't have DNA at all. They have RNA as their genetic information, and they first make DNA from their RNA, and then that DNA gets turned into proteins. So, well, that DNA gets turned back into mRNA, which gets turned into proteins. So it's not 100% clear that RNA or that DNA is the first molecule. Plus, we already learned a few minutes ago that to make DNA molecules, you need to start off with RNA molecules. So the discovery in 1990 that RNAs could play a role in catalysis, a function once thought to be just protein domain, was followed by several more discoveries about so-called ribozymes, or enzymes that functioned as RNAs that had enzyme activity, functioned as enzymes. This suggested the answer to a long-standing chicken or egg puzzle, right? If DNA encodes proteins, but the replication of DNA requires proteins, then how did a replication system even start? This could be solved if the first replicator was RNA, a molecule that can both encode and carry out catalysis. So this idea was called the RNA world hypothesis, and it suggested that DNA as a genetic material and proteins as catalysts came later, and eventually prevailed because of the advantages that they offer. The lack of the 2' hydroxyl on the deoxyribose makes DNA more stable than RNA. The double-stranded structure of DNA also provides an elegant way of replicating it. RNA catalysts, however, remain as a remnant of that early world. So we still have some of the leftover molecules that were catalytic, RNA molecules, even though peptides have largely taken over the role of catalysis. Even the formation of peptide bonds, which is essential for the synthesis of proteins, is catalyzed by RNA. Okay, so let's what was I going to go over next? I think I was going to go over secondary structure. So let's talk about secondary structure. So with respect to structure, RNAs are more varied than their DNA sort of counterparts, created by copying regions of DNA. That's how RNA comes around. Cellular RNAs are synthesized as single strands, but they often have self-complementary regions, so they can complementary bind within their own structures to each other, leading to what we call foldbacks, where they actually do have some duplex regions like DNA. So here in a tRNA, this is typically shown as the that was going to say L form, because it kind of looks like an L, I guess, upside down. This is the clover leaf representation of a tRNA. But you can see it more in the tRNA cloverleaf, these little regions of duplex, these secondary structures. Self-complementary in that they self-bind to each other. So these foldback regions, you can see that in the RNAs and the tRNAs, some RNAs, I've got some microRNAs here as well with what we call stem loops. We've got, let me just list them. So we've got tRNAs, transfer RNAs. These are for bringing amino acids into the growing peptide chain. We've got mRNAs. These are synthesized from the DNA as an intermediate in transcription. This has the information for the amino acids in the formation of peptide. We have rRNAs, or ribosomal. A ribosome is kind of a ribozyme in that it's a protein with a RNA portion in it. This is also going to be involved in protein synthesis. We have small nuclear RNAs, SN RNAs. We have microRNAs. We have, let's see, what are the snRNAs? snRNAs, small nuclear RNAs. These process initial mRNA into its mature form. So they're involved in processing or cutting out introns. Our genes have non-coding sections in them called introns that have to be taken out. There are microRNAs, MI. Like these are two microRNAs. MicroRNAs often play interference. They run around and just kind of pair up with mRNAs that we don't want our bodies overproducing. They can even run around and kind of protect you from viral infections. If a virus goes into your cell, starts dumping all its mRNAs around, your little microRNAs are a way of protecting against that. They go and they bind to those mRNAs and they prevent them from being transcribed. Transfer RNA, ribosomal RNA, messenger RNA, small nuclear RNA. Small interfering RNAs are like microRNAs. They also are used to knock out genes and to they affect gene expression. And then there are long non-coding RNAs. It's interesting. Most of the DNA in your cell is actually RNA floating around like that. Or actually, that's incorrect. Most of the RNA in your cells are interfering and microRNAs. Okay. What else do I want to say? tRNA cloverleaf here. We'll get to it eventually, but this just in case you're curious, these tRNAs carry the amino acid here at the 3' side. I don't know if I have a picture of a ribosomal RNA. I do. Excellent. Okay. Oh, I had this picture. That's funny. Okay. So I said all of this. Let's talk about what the ribosome does. So a couple of ribosome pictures here. So first off, ribonucleic acid found in ribosomes, site of protein synthesis. Only a few types of RNA, rRNA, exist in the cells. So this is rRNA. It accounts for 60-65% of the total weight of the ribosome in terms of the RNA, and then the protein portion is about 35 to 40% of the weight. In both prokaryotes and eukaryotes, ribosomes often have two subunits, one larger than the other, and so I'm kind of showing you that here. So in our whole cells, we've got these big ribosomes, but they are often made up of multiple subunits. We find these out by doing some kind of centrifugation. What kind of centrifugation is it? It's the ultra centrifugation. And then the labels there, 30S and 50S, obviously those don't add up to 70S. It has nothing to do with real math. That has to do with just the sedimentation rate or something like that. So yeah, these different subunits, they vary from prokaryotes to eukaryotes. We have different subunits than they have, but most of these ribosomes do come in subunits. Now, one is larger than the other. Okay, let's see. Typical structure and a version there. I don't know if that's bound to something. I think that might actually have something bound to it in the middle, that little red bit there. Can't remember. That's the structure of the 50S ribosomal subunit. The rRNA is shown in brown and the active site is in red. So rRNA is in brown, protein is in blue. So you can see it's mostly rRNA. All right, what was I gonna say about this picture? I guess you can just see here how ribosomes will work in our bodies. So in our bodies, as the gene is being transcribed into mRNA, ribosomes just run along the mRNA and actually start making proteins. And so you can see here, there's a ribosome running along the section that would be gene A, making A protein, polypeptide A, and then as it gets to the section of gene B, it starts to make B polypeptide, and gene C polypeptide. In DNA, in prokaryotes, the ribosomes, many of them at one time can be running along this mRNA, and also all of the genes are just sort of encoded into a big long sequence on the mRNA. In eukaryotes, it's a lot more complicated. We have exons that code for protein and introns that don't, and so before we can let ribosomes do their job, we actually have to do some processing. We have to splice out the introns, we have to cap the ends of our mRNAs with polyadenylated tails and methylated 5' caps to protect the ends from degradation. And then after the splicing happens, the exons are connected, and then we pump this out of the nucleus so that a ribosome can come and make a protein out of it. We're going to go over all of those processes in the next chapter or in the next lecture, but just at a glance here, that's a little bit of how they're different. Now I mentioned that RNA is less stable. Let's talk about base pairing for a moment. So base pairing. So RNA is slightly different than DNA, just as a reminder. Uracil is in place of thymine, so we get U pairing with A and still G with C. Like thymine, the two hydrogen bonds, but unlike thymine, uracil can actually, to a limited extent, base pair with guanine, which actually gives rise to more possibilities for pairing within a single strand of RNA. So for some of the folding. And those additional base pairings mean that it can fold in ways that single-stranded DNA cannot. So, of course, folding is critical. We know that for protein function. Like proteins, RNAs can be catalytic, and so they're going to need to have proper forms or folded forms as well. Let's see. As far as stability goes, I mentioned that the 3' deoxyribose, or the deoxyribo 2' carbon is a lot more stable. So the presence of that 2' hydroxyl makes the ribose more prone to hydrolysis than DNA does as far as the deoxy version goes. And then as far as uracil goes, since RNA has uracil instead of thymine, it turns out the cytosine is the least chemically stable of the nucleic acids, and it can spontaneously deaminate and turn itself into a uracil. So this reaction occurs in both DNA and RNA, but since DNA normally has thymine instead of uracil, the presence of uracil in DNA just indicates that deamination of a cytosine has occurred, and the uracil just gets replaced with a cytosine, like your body's smart. But in an RNA molecule, it's the bodies can't tell very easily if a cytosine turned into a uracil or if that uracil is supposed to be a uracil. And so mutations in RNA have much fewer consequences than mutations in DNA because they don't actually get passed between cells. RNA is also quickly turned over, so we make an mRNA, we make a protein from it, and then we degrade that mRNA. Whereas your DNA is kind of a long-term storage molecule. So you don't, it's good that we don't have uracils in DNA because our mutations might actually go up, or the possibility of incorrectly putting. So what I mean is, if in DNA there's a uracil, what are we going to pair against a uracil? Well, we're going to know that uracil was actually a cytosine, so we're going to pair it with a G. But in RNA, is that U supposed to be a U, or was that supposed to be a C? And when we're putting the correct amino acid in, that's important. Of course, when we get to translation, we'll see that our body has a second genetic code, basically some redundancy or degeneracy that's built into the codes that make it so that some substitutions or mutations actually give you the same amino acid. So no harm, no foul. Lastly, catalysis. So RNA structure, like protein structure, has some important, and in some cases, important regions for catalytic function. So like random coils in proteins that give rise to some tertiary structure, and There's some single-stranded regions of RNA that actually link duplex regions and give these molecules a tertiary structure. So catalytic RNAs called ribozymes catalyze important cellular reactions, including the peptide bond formation in ribosomes that we talked about. DNA, which is usually present in cells already in a duplex form, is not known to have any role in catalysis. RNA structures are important for other reasons. In addition to catalysis, the 3D arrangement of the tRNA is actually necessary for enzymes that attach amino acids to them to do so properly. So it's folded that way so that the enzyme, the tRNA synthetases, can actually work with them. Small RNAs, the small interfering RNAs found in the nucleus of cells, they appear to play a role in both gene regulation and in cellular defenses against viruses, as I was kind of mentioning. And the key to these actions, if I jump back and show you that little interfering RNA here, or this is like one, some of the key to the mechanisms is the formation of these short fold-back regions that are actually recognized by cellular proteins and then chopped into smaller units. And then one strand is copied and used to base pair with a specific mRNA to actually prevent the synthesis of that protein. Again, part of a system for defense, part of a system to keep foreign DNA from being transcribed, and the end of the chapter here. Now, I think your book does have a little bit more on CRISPR, on using Cas9 technology. CRISPR you can read about in the book. I don't have any good images, and plus I've never used CRISPR. When I was doing mutations and inserting plasmids into bacteria, we were using a form of mutagenesis. So we were just causing random mutations, and then we would go and sequence the DNA to see what happened, as opposed to what CRISPR does is you write out these codes that you want to, you program the DNA specifically, and then you go and you can insert it into the genes. It's really neat technology. I just don't know very much about it, so I don't want to say a bunch of stuff that I don't know about. But your book does have some information on CRISPR, and if you guys have any information on CRISPR that you think might be useful to me going forward, please send that to me. Alright guys, that will do it for this lecture. I got to do the next one now, so I will see you in a few.